Good day, everyone. I'm François Rousseau from Quebec City, professor at the Faculty of Medicine of Université Laval, and I'll be presenting on improving the fetal fraction of EDTA-Gel and NIPS sample using gel-based size selection. This research team is a research team from our lab here in Quebec City, but is part of a much larger project called Pegasus 2, which is funded by Genome Canada, Genome Quebec, and a lot of other organizations that I will mention later. Learning objectives for today's talk are first to understand the importance and impact of fetal fraction on non-invasive prenatal screening assay performance, and second, to understand how gel-based size selection must be applied to enrich maternal sample in circulating cell-free DNA fragments of fetal origin. This project, as I mentioned, is a very large multi-investigator research program working in the field of prenatal screening using genomics methods. Major funding comes from Genome Canada, Canadian Institutes for Health Research, Genome Québec, Genome BC, Fondation de l'Université Laval, and the CHUQ Québec Université Laval. But also the project has some in-kind contributions from Illumina Canada, Eurogene Health, Thermo Fisher, QIAGEN, and other organizations, either public or private. So the field of prenatal screening for fetal aneuploidy has been revolutionized by the occurrence of two different discoveries, one discovery and one technology. The first is the discovery in 2007 by Dennis Lowe of the presence of DNA, small fragments of DNA of fetal origin in maternal blood, in the plasma component of maternal blood. This combined with the very high throughput capacity of next generation sequencing for mapping millions sequencing in parallel and then mapping millions of DNA fragments randomly or in the targeted fashion. This combined to get those two phenomena combined together allowed for a while the development of non-invasive prenatal screening, which is a method which allows to very with high efficiency, sensitivity and specificity identify extra chromosome in the fetal compartment. So plasma, as you know, represents roughly 50% of the volume of a blood sample, the rest being white blood cells for 1% and red blood cells for 44%. The discovery of Dennis Lowe that there were some small DNA fragments in circulation in maternal blood, some of which about 15% come from the placenta and can inform about the fetus's genome, is an interesting phenomenon. However, the concentration of this circulating cell-free DNA is very low, and there are different ways to sample maternal blood to have access to this precious material. First is the use of cell-stabilizing sampling tube, which is the most currently used approach. Those specialized tubes are expensive. However, they allow to stabilize cells at least 7 days at room temperature and sometimes up to 14 days, and thus it prevents plasma contamination by genomic DNA coming from maternal white blood cells. We published the possibility to use EDTA-Gel sampling tubes as well to as long as the cells, white and red, are separated from the plasma within 8 hours from sampling to provide an IPS grade plasma after filtration. As you know, the principle here is it's explained by this schematic. On the left slide, you have left side of the slide, you have a nucleoid fetus, maternal DNA plasma fragments, of which there are some from all the different chromosomes, of course, and some from maternal origin in pink and blue, and some from fetal origin in green and red. And On the right side, you have aneuploid fetuses, and you will see that roughly you have the same representation in proportion of all the types of fragments of all the chromosomes. However, if the fetus has an extra 21 chromosome, we will be able to find extra fragments of DNA in maternal plasma coming from that chromosome, and the relative representation or chromosomal ratio of those fragments to the total number of fragments will be slightly increased. Next generation sequencing allowed us to count several millions, between 4 and 10 million of those fragments randomly, and then map them on the human genome and calculate the chromosomal ratio, which is the number of reads from a given chromosome divided by the total number of reads. And then this can be normalized to compare all the chromosomes together on the same scale, which is shown here. And for an aneuploid pregnancy, you will see that all the ratios come within plus or minus 3 standard deviations, or within the Z-score of plus or minus 3 from the mean of the ratio of the chromosome. In the case of a fetus which has an extra chromosome 21, or Down syndrome, or trisomy 21, we'll see a clear increase in the number of reads coming from chromosome 21 as compared to what would be expected from the other chromosomes, and then we will be able to measure this discrepancy in the number of multiple of the standard deviation or the Z-score, which will provide us a signal that there's a likelihood, significant likelihood of having a fetus which has an extra chromosome 21 in this specific case. This slide illustrates the results of several thousand analysis of the sort which have been done in the Pegasus-1 project, and it shows that on the x-axis you have the fetal fraction, on the y-axis you have the Z-score, and you have the final call of the pregnancy. So the red dots are all fetuses which have confirmed trisomy 21, and the green dots are fetuses which have a normal karyotype. And then you see that below 4% fetal fraction, it may be difficult to distinguish between true negatives and false negatives. So usually the labs will have a minimal fetal fraction to call a result in our case, and most of the labs use a 4% cutoff. The fetal aneuploidy increases the representation of the affected chromosome, but this measure has a certain is not perfect like any lab measure. It has some uncertainty, and the more precise the measure is, the more easy the more easy it is to separate cases of fetal aneuploidy from aneuploidy, and the larger the fetal fraction on the right side of the right graph, you see a 12% fetal fraction case, and on the left side of the slide you had a 4% case. So the more fetal fragments you have in the maternal component of the plasma, the larger the Z-score, and the more sure you are of the result. Higher fetal fraction also yields higher chromosome ratio scores, but as I've shown, better discrimination, and this is shown here. So as the fetal fraction increases on the x-axis, you see that the distance between cases and euploid fetuses, cases are the bold filled line, which is raising while with the fetal fraction, while the dashed bold line is horizontal and doesn't change with fetal fraction for euploid fetuses. So, and you have plus or minus 3 standard deviation illustrated on each side of those lines. So you see that the larger the fetal fraction, the more distance there is between cases and controls. Fetal fraction is also more frequently below our minimal level in the early pregnancies, as shown here. It is also lower in female in pregnant mother, which have a high body mass index, which is shown here. Improving fetal fraction or monitoring fetal fraction is a very important component of performing a non-invasive prenatal screening test. Here is a slide which summarizes the pros and cons of the different blood sampling alternatives. So you have on the first line the specialized cell-stabilizing collection tubes, such as STREC tubes. They are roughly tenfold more expensive than the EDTA collection tubes, either regular or with gel. They are more stable. However, you cannot isolate an IPS grade plasma at the site of collection usually, because it asks for high-speed ultracentrifugation. They have to be shipped at 6 degrees or more, so they don't really tolerate low temperatures. And the burden to an IPS lab is more important than receiving an EDTA collection tube, especially a gel tube, for which you can simply decant the plasma just by flipping the tube into another one. We published that you can filtrate this first step plasma and you then obtain an IPS grade plasma. This slide shows you the pictures of the procedures for using EDTA gel tubes for an IPS. So after sampling, they are centrifuged. The plasma is recuperated on top of the gel, while the white and blood cells are below the gel. And we simply filter using a specific filter into the another tube, and you have an IPS grade plasma that can be shipped to the lab, even frozen. So some countries have to ship IPS samples for long distances, and it's frequently the case because non-invasive prenatal screening labs are very specialized and are expensive to set up. They need to be very well acquainted by the method, so it's frequently the system is frequently with a centralized lab, and samples are shipped to the centralized lab from more or less distant parts where women are sampled. So those samples need to arrive at the lab, making sure that the plasma has not been contaminated by genomic DNA from white blood cells during transportation. So ideally, they must be able to travel long distances, especially in countries where you can encounter harsh temperature conditions, such as Canada, where we have even received frozen samples that came from different areas north of the country. So we published this validation of a protocol using EDTA-Gel samples and filtration, which provided the same yield of circulating cell-free DNA, the same level of fetal fraction as STREC tubes when compared. So you can read that paper if you're interested. And in our studies in Canada, we have been shipping samples for a long time now between Vancouver and Quebec City, which is a 4,800 kilometer distance. The tubes can be centrifuged. In this present study, the tubes were centrifuged within immediately after collection, within 6 to 8 hours, and then kept at 4 degrees, sent to the central lab after 2 to 5 days in batches. And this has worked to identify method to rescue such samples, because in some instances those samples arrived with a very low fetal fraction. So we attempted to search method to recuperate circulating cell-free DNA, if possible, of use for an IPS, and we postulated that using gel-based selection could be a promising approach for this. As you know, most likely, most of you, fetal fragments in maternal blood are smaller than maternal fragments. This has been known for a while. This was a paper published in 2004, again by Dennis Lowe's team, showing in blue the size distribution of the concentration of fragments of different sizes coming from the fetus in blue and mother in red. So you see that maternal fragments are much larger. This was using two genes, one which was specific to male fetuses on the Y chromosome for the blue, and leptin as a maternal marker. So the research question of what I'm showing you today is how can we compare EDTA-Gel sampling using size selection to STREC sample relative to the quality of cfDNA for an NIPS analysis? So we compare 61 samples collected in Vancouver in both EDTA-Gel tubes and STREC tubes and shipped over to Quebec City for testing by NIPS. This sample size allowed us to detect a 1% difference in fetal fraction between the two groups. In terms of methods, all the women were between 10 and 14 weeks of gestation. We took 2 samples of STREC tubes, one of EDTA-Gel. EDTA-Gel tubes were centrifuged and sent within 6 hours and then shipped, as I mentioned. STREC tubes were shipped by overnight twice a week to Quebec City. Shipping occurred as planned. However, for 2 shipments of EDTA-Gel samples that were not with STREC gel samples, they were delayed 4 to 5 days on top of the 9 days that usually took at maximum to get to the central lab. So those samples allowed us to test what happened and then to find a way to recuperate those samples at some time using gel-based site selection. So this is the details of the NIPS method. So STREC tubes were centrifuged according to standard protocols, EDTA-Gel tubes as well, as I've shown. cfDNA was prepared using QIAGEN kits, and next-gen sequencing libraries were prepared with a Kappa kit from Roche. Libraries were quantified and pooled for sequencing, both for STREC and EDTA-Gel tube, but a second step was added for the same batch of EDTA-Gel libraries, which was sample selection using a light-pitch instrument from Neurogene Health, in which we used 25 microliter of sample and on a 3% precast gel. Software was selected to extract DNA ranging from 229 to 286 base pairs. There's a typo here, which corresponds to insert size between 100 and 160 base pairs, roughly what we expect for the fetal fragments to fall within. Paired sequencing was performed, also allowed us to calculate the fragment length, and read were mapped to the HG19 reference genome. So sample characteristics are shown on this table, and you see that the mean gestational age, the weight of women going up to 130 kilos, body mass index up to 42, and we had 19 women with a higher body mass index at 25. This is a very interesting table summarizing our results. So you have a column for the STREC tubes, which were used according to the standard protocol, which yield the fetal fraction, mean fetal fraction of 8.6%, with a range of 2 to 26. There were 7 samples below 4%, or 11% of all the samples here that were below our cutoff for reporting at the first pass. Of course, we usually retry to re-extract and resequence, and we can save several of those samples, but there are always some remaining for which we can't report a result. The EDTA-Gel tubes, there were 47 which were not delayed, and 14 that were delayed and arrived, as I mentioned, up to 5 days later than the rest of those tubes. So for the no delay samples, some prior to size selection, we had a lower fetal fraction than strect tubes, and we had 9 women with below 4%. After size selection, the mean fetal fraction was much improved and even larger than for strect tubes at 13%, and none was below 8%. So it was very an interesting result. And for the delayed samples, the fetal fraction before size selection was 4% for the same samples of the same women, and 50% were below the cutoff of 4%. But after size selection, again, the fetal fraction was even higher than for strect tubes, and there was no sample below 6% fetal fraction, so they were all usable for an IPS. A couple of other results shown with respect to here the number of days after a blood draw. So you see in green the strect tube fetal fraction, in yellow the EDTA fetal fraction, and in orange the after size selection for the EDTA-Gel fetal fraction. So you see that in general they're always improved, and in fact the lower the fetal fraction from the start, the larger the increase after size selection. Here is just the individual results, and you have the delayed samples with blue stars below their numbers and on the x-axis, and you see that the size selection EDTA tubes is almost always above the others, except for some samples which have been delayed, for which in a couple of occasions the strect tubes yield a higher fetal fraction. In order to see if this would reflect into the Z-score, we looked at the Y chromosome of the male fetuses, and indeed there was a significant improvement of the chromosomal ratio, thus Z-score of the Y chromosome, suggesting that the same improvement will be seen for aneuploid fetuses as well. In terms of the size distribution of the fragments, this slide shows you in gray the size distribution of EDTA-Gel samples before size selection, and in blue after size selection. So you see that the system is very efficient at removing virtually all fragments above 160 base pairs, and the fetal fraction is much improved, and we get rid of most of the maternal fragments which are more concentrated in the right part of the graph. Sorry. This is the distribution of the strect tube. As you see, it's totally superposable to the distribution of the EDTA-Gel tube before size selection. Here is a graph which shows you the difference between the delayed and undelayed EDTA-Gel tubes. So delayed are in red, undelayed are in gray before size selection, and after size selection the delayed are in yellow and the undelayed are in green. So you have the same efficiency of size selection, and you see that the trail of the delayed samples in red is slightly towards the right with a lower peak, but all those fragments are removed by size selection. According to the BMI here, you see again similar phenomenon, again gray streak, yellow EDTA, and orange EDTA size selected. And again, you see the increase is almost, the size selection process almost doubled the fetal fraction of EDTA-Gel samples for high BMI women. So, if we summarize, the expensive cell-stabilizing NIPS collection tubes are frequently recommended to maintain white blood cell integrity and protect cfDNA from degradation over several days. They must however, be kept at 6 degrees or above. We've shown that the EDTA collection tubes offer an alternative where an NIPS-grade plasma can be produced at the collection site and shipped frozen. Alternatively, they can be also shipped after low-speed centrifugation simply without separating plasma from the gel and the tube content below the gel. Gel-based cell selection has been shown by others to have the capacity to increase fetal fraction, but it has not been applied to those types of sampling tubes up to now. We show here that the use of gel-based cell selection increases the fetal fraction in those samples collected in the EDTA-Gel and shipped after low-speed centrifugation. We also showed that gel-based cell selection of NIPS libraries can rescue samples that have been contaminated by maternal genomic DNA. Gel-based selection, cell selection in the present study achieved higher fetal fraction than STREC tubes using the standard protocol. And our results also suggest that the cfDNA integrity is maintained in EDTA-Gel collection tubes and can then thus be rescued by gel-based cell selection. The main limitation of this study is that we didn't have any case of aneuploidy in the sample of 61 women, which was collected prospectively. However, our analysis of the Y chromosome ratio and its Z-score suggests that the other chromosomes will also be enriched. And this has also been reported using cell selection and other types of collection tubes as well for aneuploid chromosomes. So in conclusion, cell selection of NIPS libraries significantly increases the fetal fraction of EDTA-Gel collection tubes for NIPS. This yields to improved clinical performance of all NIPS assays and lower false positive rate and lower false negative rates. Cell selection also has eliminated in our hands all NIPS failures due to low fetal fraction. So this is expected to reduce significantly the number of withdrawals for women with high BMI or early pregnancy samples. Cell selection also rescued NIPS samples that have been contaminated by maternal genomic DNA, so again, should lead a lesser need to redraw due to low quality sample. And finally, cell selection of EDTA-Gel samples may be very useful for enrichment of fetal DNA in the context of multiple pregnancies, screening of subchromosomal imbalances, and probably also to increase the proportion of tumor cell-free DNA in liquid biopsies for oncology. So we thank you very much, and we hope that you found this presentation interesting.